Previous coverage of the immune response have been all about the beneficial aspects, which are clearly protective to us. But another side of it is that this system promotes rather than prevents disease. In this chapter, we're going to be studying immunopathology, which is the study of disease that results from either an overactive or an underactive immune response. The immune system has to be carefully controlled. If we have too little expression, we'll get sick from pathogens. But if there's too much expression, we can damage our own tissues. So immunopathology is the study of disease states that's associated with overactivity or underactivity of the immune response. In this pink area here, we see a region where we have overreactivity. This section involves things like allergy, hypersensitivity, and on the extreme, autoimmunity, where our immune system attacks our own cells without any particular antigen being present. Here, the tissues are innocent bystanders, but they're attacked by the immune system because the immune system is unable to tell self from foreign. In immunodeficiency diseases, shown here in the blue region, immune function is either incompletely developed, it's suppressed, or becomes destroyed, for example, by AIDS. Cancer is in a special category all on its own because it's both the cause and effect of immune dysfunction. For example, we could lose surveillance, which causes cancer, and that cancer could then cause a loss of immunity. In this chapter, we'll be covering several types of overreaction systems that involve antigens. As you can see here from the pink area, there's type 1 overreaction, type 2, type 3, and type 4. At a basic level, we can look at types 1, 2, and 3 as involving B-cell immunoglobulin responses. Type 1 is for common allergies and anaphylaxis. In type 2, we have antibody-mediated cell damage. And in type 3, it's an immune complex of both humoral and antibody. The type 4 is a more delayed overreaction. This involves the T-cell response. The table here will show you some examples that are associated with each of these different types of overreactions. The antigens causing overreaction can be exogenous, like pollen or microbes or grains, or endogenous in the case of autoimmunities. Allergies are often mistaken for infections because both systems involve damage to the tissues and thus trigger the inflammatory response. So there are very similar symptoms between infections and allergies. So let's begin by taking a look at type 1 allergic reactions. These are allergies and hypersensitivities. People who are allergic have acute sensitivity to repeated contact with antigens. So we call these allergens because they do not affect any non-allergic individuals. So an allergy is an altered reactivity or exaggerated immune response manifested by inflammation. Hypersensitivity is sometimes used interchangeably with allergy, but some professionals consider this to be a delayed reaction. So while allergies are immediate, hypersensitivities take some time to onset. The allergen itself is an antigen to which allergic individuals are sensitive, and an immune response is elicited. There are two levels of severity of type 1 allergies. There's atopy, which is our chronic local allergy, like hay fever or asthma or food allergies. Then there's anaphylaxis, which brings on anaphylactic shock. Usually this is systemic, and often it can be a fatal reaction. Either way, the reactions that we see in allergies and hypersensitivities include all of those we saw in the preceding two chapters: the humoral response, the cell-mediated response, the inflammatory response, phagocytosis, and use of the complement system. So this is going to be a great way to review that material. So the majority of type 1 allergies are relatively mild. Some, like anaphylaxis and asthma, are a little more serious and require hospitalization or could actually cause death if not treated. And predisposition for type 1 allergies seems to have a really strong familial association. This familial association is a generalized susceptibility, meaning a generalized potential to have allergic reactions, not a specific allergy. For example, if a mother has hay fever, then the child may have some other sort of allergy. This hereditary link for the propensity to have allergies in general seems to be a genetic program that favors the allergic antibody IgE production, as well as an increased reactivity of the mast cells and an increased susceptibility of the target tissues. About 10 to 30% of the population seem to suffer from allergies. Now, many of these could be unreported due to the availability of over-the-counter remedies. Age, previous infection, and geographic locale also will determine the risk for allergies. There is a hypothesis called the hygiene hypothesis that shows that cesarean delivery has a high correlation with the development of allergies when the mother herself was also allergic. So having the combination of a cesarean delivery and an allergic mother increases the risk factor to 8 times that of a non-cesarean delivery. Now it is key that both of these conditions exist, cesarean and an allergic mother. It's understood that the baby needs the exposure of the commensal bacteria that it would experience in the birth canal for development of the IgA system. If it doesn't get this experience, then IgE is predominant, and thus we have a propensity for allergic reactions. Researchers have also noticed that breastfeeding for at least 4 months vastly decreases an infant's chance of developing allergies. This is proposed that the cytokines and growth factors that are found in breast milk induce tolerance rather than reactivity to allergens that pass through the gut. As with other antigens, allergens have certain immunologic characteristics. You'll recall that in general, antigens that are proteins are more allergenic than those that are not. For example, than carbohydrates, fat, or nucleic acids. You'll also recall that these protein molecules needed to be larger and more complex. Some allergens are haptens. These are the very small ones that are non-protein, but they need to have a carrier molecule. Allergens can also be organic or non-organic molecules in household products, cosmetics, food, or drugs. The table below will give you a good overview of some common allergens that are classified by their portal of entry. We can have inhalants, ingestants, injectants, things that are injected either by a needle or by an animal, or contactants that actually need to touch the skin. Now, the organ of allergic expression may not actually match the portal of entry. For example, you could have something that enters through the gastrointestinal portal that manifests itself as a rash in the skin. Typically, allergens enter through epithelial portals in the respiratory tract, GI tract, and skin, just as many of the microbes would enter. So again, inhalants are things like house dust, dander, and pollen. They're airborne environmental allergens. Allergens that are entering by the mouth are called ingestants because we ingest them. They come into the GI tract. Sometimes there are injected allergies. Some of these are side effects of drugs or other substance that are used in diagnosing, treating, or preventing disease, but they could also originate through venom from things like bee stings. And allergens that enter through the skin, again, are called contactants. So why do some people experience allergies and huge symptoms associated with them, while other individuals have no symptoms whatsoever? It really comes down to what occurs in the tissues of an allergic person. These symptoms don't happen in the tissues of a non-allergic person. Type 1 allergies occur in stages. There's a first exposure and a second exposure. Generally, the first exposure elicits no sort of signs or symptoms. You may have heard of people that are allergic on their first exposure. However, it's fairly well understood that it takes multiple exposures, and thus the individual, probably unbeknownst to them, did have a first exposure with no actual response. I think of it as recently I came up as allergic to bee stings, but I don't remember being stung by a bee, although perhaps once when I was a child I was stung by a bee. So I was already sensitized. The sensitizing dose is the one that's going to prime the immune system for any subsequent encounter. It elicits no signs and symptoms. However, memory B cells are produced. This makes them ready for the reaction of the immune system when there's a subsequent exposure. The mast cells and basophils are also equipped with these IgE antibodies. Let's take a closer look. When an allergen penetrates its particular port of entry, the lymphatics will carry allergen particles to the lymph nodes. In the lymph nodes, the clones of B cells that were all generated will recognize the allergen. That specific clone will be activated and proliferate into plasma cells. The plasma cells then produce IgE antibodies. These are the antibodies of allergy. Remember that there's a constant region and a variable region. The variable region is the one, again, that's specific to the particular antigen that's being presented. However, the constant region is the stem, so this portion of the antibody. And that stem has a great affinity for mast cells and basophils. So the IgE will bind itself to the mast cell surface receptors. Here's a mast cell with many of those IgE antibodies, or IgE receptors, bound to its surface. The binding of these IgEs to the mast cells causes the reactions that occur upon repeat exposure to the allergen. So how do the mast cells work in this case? Well, the mast cells and basophils are ubiquitous in tissues. That means they're located in connective tissues of virtually all organs. But there are exceptional numbers that we find in the lungs, on the skin, on the GI tract, and the genitourinary tract. Now you'd expect this because these guys are ready for infection, so they're just where we need them at all of the portals of entry. Now these mast cells have a high capacity to bind IgE during the sensitization phase. In fact, each mast cell can bind 10 to 40,000 IgE antibodies. And each mast cell is packed with granules. These granules that you can see inside the cell contain physiologically active cytokines like histamine and serotonin. And they also have a tendency to degranulate. So upon the second encounter of this allergen, the allergens bind onto the IgE receptors, which causes degranulation or release of all of these different cytokines into circulation. So it's not until the second contact with the allergen that these IgE primed mast cells release all of their granules or undergo degranulation. These IgE primed mast cells can remain in the tissues for decades, and the person then retains the capacity to react immediately upon re-exposure because of the degranulation. Next time the allergen molecules contact the mast cells, they bind across adjacent receptors, which stimulates degranulation. These chemical mediators like histamine are released and diffuse into the tissues as well as the bloodstream, and they give rise to local as well as systemic reactions, such as our red and itchy eyes or hives, or a runny nose, perhaps from hay fever. Let's take a look at a quick animation that shows type 1 hypersensitivity reactions.
Some people develop an allergic reaction or hypersensitivity when exposed to substances such as dust, pollens, animal dander, or penicillin. This hypersensitivity is mediated by IgE. Sensitization occurs when the antigen makes contact with some part of the body. The antigen is taken up, processed by antigen-presenting cells, and presented on a class II MHC to T helper cells. Tissues under the mucous membranes are rich in B cells committed to IgE production, and IgE-producing cells are more abundant in persons susceptible to allergies. The T helper cells produce cytokines which stimulate these B cells to proliferate and differentiate into IgE-producing plasma cells. As IgE is produced in specific areas of the body, the IgE molecules attach via their constant regions to receptors on nearby mast cells. Mast cells contain granules packed with chemicals that induce a hypersensitivity response. Once attached, the IgE molecules can survive for many weeks. The individual is now sensitized to the antigen. When exposed to the antigen for a second time, the antigen binds to the IgE antibodies on the mast cells. To trigger a response, two cell-bound IgE molecules must react with a specific antigen. Within seconds of the reaction, the mast cell releases histamine and other mediators of the inflammatory response from the granules, triggering a variety of symptoms.